In this video, we're going to look at the structure of proteins and the biochemical test for proteins. All proteins are made of amino acids. These are the basic building blocks that make up our polypeptide chains. They are joined together in a series of condensation reactions where water is lost and they form a peptide bond. Your general structure of an amino acid looks like this. At this end, we have an amine group, so NH2. At this end, a carboxyl group, COOH. We have a hydrogen up here and an R group down here. Now, in general, this is the only bit that changes in your amino acid. So here we can see hydrogen, so that is your basic amino acid, glycine. In general, there are 20 different amino acids that are common across all organisms, and they only differ by that side chain or R group. Now, you don't need to know the structure of any of these amino acids. You just need to know that the R group is what determines the properties of the amino acid. So this could be polarity, it could be the presence or absence of sulfur, or something like charge. Amino acids join together and form a peptide bond. The peptide bond forms between the carboxyl end of one amino acid and the amine end of the other. We lose hydrogen and oxygen to form water, and your peptide bond looks like this. It forms between the carbon and the nitrogen, and it's shown here in green. This is the bond that forms from the condensation reaction between your amino acids. And the peptide bond can be written as CONH. Proteins have three basic layers of structure, starting with the primary structure. We then move up through the secondary and the tertiary, with more complex proteins having a quaternary structure. Your primary structure is basically the order or the sequence of amino acids joined together by peptide bonds. This long chain is called a polypeptide. Now, it's ultimately this primary structure that determines the overall characteristics of a protein, and a change in one single amino acid could alter the entire shape and therefore the function of the protein. This could lead to it becoming non-functional. It's usually represented by this long chain, with our circles representing different amino acids and the lines between the peptide bonds. The next layer of protein structure is called the secondary structure, and this is formed by hydrogen bonds. Okay, they form between the H of the amine group and the O of the carboxyl group, and this coils up to form an alpha helices or a beta pleated sheet. So if we take our primary structure in a chain, this then can coil up into what would be represented as an alpha helix, okay, which is a spiraled shape. It could also form this more kind of corrugated effect for our beta pleated sheet. These are all formed by hydrogen bonds, and they make up the secondary structure. Our next level of protein structure is the tertiary structure. This is formed when the alpha helices and the beta pleated sheets twist up and fold up further. This gives our proteins a specific 3D structure. The structure is maintained by three types of bonds. We have disulfide bridges, ionic bonds, and hydrogen bonds. Our disulfide bridges are formed between sulfur on the R groups, ionic bonds between opposite charges, and hydrogen bonds between free hydrogen and oxygen molecules. Here we have a representative diagram to show those chains now twisting up to form a specific 3D shape. The final stage in our protein structure is the quaternary structure. Now, not all proteins have this, but this is when there is more than one polypeptide chain joined together. They often have non-protein prosthetic groups associated with them, and hemoglobin is a really good example of this. Hemoglobin has 4 polypeptide chains with an iron-containing heme group in the center of the chain, and it looks a little bit like this, with these yellow circles here representing your ferrous group. This is how hemoglobin carries oxygen around the body. Now that we've been through our protein structure, let's summarize. So we start with our order or sequence of amino acids in your primary structure, held together by peptide bonds. This coils up and creates your alpha helix, held together by hydrogen bonds. We then coil up further to create our 3D structure, where we have our ionic, our disulfide bridges and hydrogen bonds, as well as those hydrophobic and hydrophilic interactions. We then move on and in some cases have a quaternary structure, where there is more than one polypeptide chain joined together. There is a really simple biochemical test that you can do to test for the presence of a peptide bond. You use Biuret solution in order to do this, and you are looking for a colour change from blue to purple. So let's go over to the lab and have a look. Now that we're in the lab, let's have a look at the test for protein. In order to do this, you need your sample. And in this case, we have some liquid albumin, so we don't have to crush this and add it to water. And we also need some Biuret reagents. This is corrosive, so if it gets on your hands, make sure you wash it off. In order to do this, we are going to add some of our sample to our test tube, and then we are going to add some Biuret reagent. We are looking for a colour change from blue to lilac or purple to indicate a positive result for protein. Okay, so in this case we have had that lovely positive lilac colour formed, which shows the presence of the peptide bonds. Now if this was a negative result, it would have simply stayed blue. So it's important to remember that you need to add that into your answer as well. And that is the test for proteins. Now that we've been to the lab, let's recap the stages. First up, we would want to grind up our substance to be tested and add some water to create a solution. Next, you're going to add an equal volume of Biuret solution and wait for a colour change. If a purple colour change happens, this indicates the presence of peptide bonds. However, if it stayed blue, then there are no peptide bonds. An alternative to adding Biuret would be to add some copper sulfate and sodium hydroxide. Proteins fall into two categories, and this will usually come up when you are looking at the application style questions. So we have globular and we have fibrous. So our globular proteins incorporate enzymes. So enzymes are globular proteins. There are others, but enzymes are the ones that you will know about. They are usually soluble in water and have a complex tertiary 3D structure. This gives them their precise 3D shape, and they are often spherical. The other type we have are fibrous. So these are things like protein, and they often have a structural function. So collagen, as an example, is formed from 3 polypeptide chains wound around each other like a rope. In general, though, they are usually insoluble in water and have repetitive amino acid sequences. Okay, so this is an example of a structural protein such as collagen wound around each other like a rope. In this video, we have looked at the structure of amino acids, the formation of a peptide bond, the primary, secondary, tertiary and quaternary structures of proteins. We have looked at the biochemical test for proteins as well as different kinds of proteins. In the next video, we will be looking at enzymes.